Good morning or afternoon to all of you. I'm not sure what time of day this video is being shown, but in either case, I'm very happy to be with you for your summer school. I'm sorry I can't do it in person, but I've heard a good deal about what you are doing, and certainly I'm very supportive of your consideration of a career starting at the university level in science, in scientific research, technology, or in related areas of what increasingly we call STEM. And what I'd like to do today is offer you sort of my perspective, which has been gained over approximately, well, almost half a century, I'm embarrassed to say, of research at the university level in basic science, in biology, and in medicine. And I believe after all of these years, I'm only more convinced that basic research is important. And what I'd like to do is share with you with a few examples from biomedical research in particular, why I feel that way. And I hope that you'll consider the importance of basic research for our societies, our lives, and our livelihoods when you consider the career path that you may take. Now, although I will emphasize today examples from biomedical research, I could equally well be talking about basic research in material sciences, engineering, computer science, chemistry, and the list goes on. My personal competence, though, limits me to talking about biomedical research. So let's begin with a fact that many of us are not aware of. This is a graph of life expectancy, human life expectancy, going back to some of the earliest recorded history. So if you go back to let's say, the Roman era, the classical era, the life expectancy at birth was about 25 years. Actually, yeah, as far as we can tell, although the records are poor, you can go back to the earliest recorded histories, and it appears that the life expectancy was no different. But if you look at this graph, you'll see, astonishingly, that nothing changed up until about here, that being the middle to the late 19th century. So what happened in the middle or the late 19th century that caused a dramatic increase of life expectancy, which now globally is in the range of 70 or more years? What happened? Well, what happened was the beginning of systematic basic scientific research in medicine in the mid-19th century. We all think of the name Pasteur, for example, or Koch, famous French and German and other European names of medical scientists who took the scientific approach, the scientific method, which was first developed, as you probably know, for the study of physics and the physical world, and then the chemical world, and took this method of thinking and of evaluating the world into the world of biology. And what they discovered very quickly was that common diseases were caused by microorganisms that were invisible to the eye, and they developed ways to measure these microorganisms and to quantify them. Methods of microbiology that we still use today, as a matter of fact. They discovered that these microbes were present in water in most cities, and that contamination of the water, and as is still the case in many parts of the world today, unfortunately, is a prevalent cause of infectious disease. They learned and taught us how to treat water with, for example, with chlorine. and how to carefully dispose of water and waste, which all big cities endeavor to do today. Health departments were instituted in various countries throughout the world, and the immunologist, soon with specific microorganisms already now associated with causes of diseases, the field of immunology could develop. And with this came the systematic approach to developing vaccines, which of course we all benefited from even recently during the COVID pandemic. And you can see here, this is now the death rate due to infectious disease as a function of years in the 20th century. You can see some of the major events, a decline in the incidence of infectious disease or in the death rate from infectious disease by chlorination of water, a simple public health maneuver. By the way, in the 19th century, the greatest advance in public health was made by Semmelweis, a Austro-Hungarian physician who discovered that microorganisms could be their contamination minimized by a simple act of hand washing. And that's, of course, saved many lives. But the development of vaccines, for example, after the influenza pandemic, influenza vaccines, the discovery of antibiotics, very importantly, during my lifetime, the Salk vaccine, as we call it, for polio, was introduced. And you can see there was a dramatic decline. So this was one of the first victories of basic medical science, the discovery that diseases are caused by specific microbes. Now, what was the consequence of this? Well, if people were dying in childhood or early adulthood as a result of infectious disease, and unfortunately we haven't completely stopped this yet, but we've certainly put a huge dent in it. If people die less from infectious disease, well, they live on. And when people live longer, what happens? Well, I think unfortunately we know what happens. We start to see degradation of the cardiovascular system, the blood vessels, the heart, the circulatory system, and with that, the development of atherosclerotic disease, strokes, heart disease, and the like. And so, perhaps not surprisingly, as deaths due to infection went down during the first half of the 20th century, now people lived longer into their 50s or 60s. And good news, life expectancy went up, as I've been saying. But bad news, people lived long enough to develop, on the average, cardiovascular disease. So what happened then is that the incidence of cardiovascular diseases, whether they're diseases of the heart, this is now again death rate, not the rate of incidence of the disease, death rate per 100,000, these are US numbers, but they are reflective of really any developed society in the world at that time. Cardiovascular disease went up, as I've described, due to the heart and also due to stroke. But then you see something else happen. Look what happened starting around 1960 through about the mid 1990s. Look at total cardiovascular disease deaths. There's an enormous decrease. It's more than a factor of 2, and by today it's actually a factor of 3. And again, this is something that has been kind of the story of my generation. My parents and people of my generation, our parents, were approaching their mid-50s in the 50-year-old or greater in 1960s, 70s, and 80s. I'm a member of the so-called baby boom generation, born after World War II. And so as we started going, when I was essentially your age, our parents were perhaps like your parents in their 40s, 50s, and 60s. And they were experiencing cardiovascular disease. I well remember, happily not my parents, but I well remember many of my friends' parents, especially the men, simply dying of heart attacks in their 1950s in their 50s and in their 60s. In fact, it was uncommon for a man to live beyond about 70. The life expectancy then was more like 65 to 70. The retirement age then as now was mostly about 65. But when a man retired and it was mostly men in those times who were employed, unfortunately, but it was the case. And when men retired and at 65, they actually had a rather short retirement on the average to worry about. Happily today, when people retire at 65 or 70 or older, they typically will live much longer than that and have an opportunity to develop new interests and new careers or to spend extended time with families. And why is that? Why is it that men, and of course women too, women have always had perhaps 5 years more life expectancy than men on the average? Never been completely clear why, but it's also true here for cardiovascular disease. So why is it that this happened? It's not an accident, actually. It's not like suddenly everything changed, the world changed in 1960, and suddenly people started getting spontaneously cured or treated for heart disease. So what happened? Well, this is what happened. We came to understand that the underlying causes of atherosclerosis were high blood cholesterol and high blood pressure. Those two factors together conspire over many years to lead to the deposition of atherosclerotic plaques at critical places in our vasculature that affect the integrity of the vasculature in such a way as to weaken it, to allow bleeding, which is what we call a stroke, particularly when it occurs in the brain, or to cause blockage through fatty cholesterol plaques that are released from where they accumulate in one part of the vasculature typically, or even can grow in situ to block our coronary arteries in the heart, or arteries the arteries that go to the brain, or other critical parts of the vasculature. And so what happened starting in the 1960s is, as the result of deep understanding of physiology, basic physiology, we came to understand blood pressure and the basis of controlling blood pressure. And we came to understanding cholesterol as a biochemical entity. We took scientists in that era undertook a study of cholesterol for its own sake. They were interested in what composed an atherosclerotic plaque. They discovered cholesterol in the plaque. And there began 50 years of study, as I'll describe now, of the basic biology of cholesterol. And what that translated into are drugs that we take today that control our cholesterol. I personally have had high blood pressure and high cholesterol. And I'm now 74 years old. I doubt that I would have lived a lot longer than my grandfather, who came from Moldova, actually, who died, unfortunately. I never got to meet him in his mid-50s. And he died in New York, but he died of a combination of high blood pressure and cholesterol, as many people did in that era. He never had the benefit of taking a drug for blood pressure or a drug for cholesterol, which I do. Now, what is the story then behind the development of these major discoveries, and how did they occur? Now, I won't have time today to go through both of these stories, so I'm going to pick the cholesterol story. But what I say about cholesterol could equally well apply to the discovery of how to control blood pressure. They're both equally important for controlling heart disease and stroke. And importantly, and this is really the major lesson today, importantly, what we learned from cholesterol applies really to any basic science program, and the impact that basic science can have, and why it requires long-term investment and a lot of patience over many years, many more than we would like to wait for these types of advances, but it's the nature of science. So where does this cholesterol story begin? Where did the drugs come from that we use today to control cholesterol and save lives on an epic scale? The story actually begins in the early, around 1920, even a little before 1920, in the wake of the recovery from World War I. And even by then, European science, especially Germanic science, in medicine was operating. When I say Germanic science, I'm referring primarily to science in Switzerland, Germany, Austria, to some extent, Hungary is part of the Hungarian Empire. And I can't remember if I mentioned Switzerland, but I probably did. In any case, these scientists, Heinrich Wieland and Adolf Windhaus, were recognized by a Nobel Prize for their discovery of the structure of cholesterol. They were just curious to know these atherosclerotic lesions, which in their time had already been associated by pathologists with heart disease. You could see patients that died from heart disease had abundant plaques. It wasn't hard to find them. And these plaques were composed of a fatty substance. What these scientists and their contemporaries did, they were chemists. They were natural product chemists, and they used their skills in analytical chemistry to describe the exact chemical structure of cholesterol. Now, and they described it as the major constituent of these plaques. So it became quickly important, and they postulated that cholesterol was important for heart disease. And they provided the first way of proceeding in a scientific basis. They reduced it to a chemical problem. They now had a hypothesis. The hypothesis is, for some reason, cholesterol accumulates in plaques. They discovered the structure of cholesterol, and now they opened the door to a new problem. We could now ask, or they could ask, how is cholesterol made in the body, and how is it absorbed from the food that we eat, so that that combination of the two could determine how much cholesterol you have in your body, and possibly even where it goes in the body, maybe even into a plaque. Well, they also discovered something else. They characterized, for example, bile acids. Now, that doesn't seem like they would have anything to do with cholesterol. They are the secretions of the bile duct of the gallbladder. And some of you may know that the gallbladder stores these compounds called bile acids, and it injects them into the gastrointestinal tract to facilitate the digestion of food. Okay? And really, what the bile acids are they're sort of soap-like molecules that act as detergents to dissolve the food, so that the enzymes in our digestive tract can eat them up and break them down to substituents, small molecules like amino acids or fatty acids, very effectively. But it turns out the bile acids are metabolites of cholesterol. They actually are simple derivatives of cholesterol, and the relationship between the two was also discovered by these two scientists. And then when those two classes of molecules were put together side by side, it could be seen that there were certain structural relationships. And that enabled, over the following years, these two scientists working in Germany and in Switzerland to come in from a completely unrelated way to discover yet another set of molecules that had something in common with cholesterol and with bile acids. And these are the sex hormones. Now, I think we've all heard of estrogen and testosterone as kind of the major so-called sex hormones that determine male or female, okay, and are important for many other aspects of biology and medicine, and are also very important in prostate cancers, breast cancers, and the like. Those discoveries actually go back to these scientists who wanted to were simply asking, were there hormones that could be isolated from the blood or the urine that were unique to males or females, and then might have a determining role? And so they discovered testosterone and estrogen, and they discovered when they analyzed the structure of these molecules, they saw that actually they had a very similar structure to the structure of cholesterol and of the bile acids. So suddenly, the world of cholesterol expanded very broadly into a world of multi-cyclic, heterocyclic chemistry of fatty substances that were related to cholesterol, steroid hormones. And they also noticed a similar structure was present, believe it or not, in rubber. Rubber that was being of great importance, being withdrawn from plants in exotic climates, and being used at that time primarily in industry for the newly expanded industry of automobile tires, and of course for many other things. And what they discovered was that all of these substances, cholesterol, steroid hormones, were made from a basic repeating 5-carbon structure that is called an isoprene. And this, I'll show you an example of that structure in a little bit. I'm trying to avoid showing you a lot of chemical formulas. But I hope you're getting the feeling that an understanding, and so I want to go back a step. The question that we're trying to ask is, how is cholesterol built in the body? The problem at that time in 1930, 1939, is that we actually didn't know how any carbon-containing molecule essentially was built, macromolecule was built in the body. We knew a little bit about how food was metabolized. For example, you've probably heard of the pathway of glycolysis. Okay, but we knew very little about how the body built up substances. So the clues were coming from many different sources. Now I'm going to take a detour, which is completely wild. It's 15 years later, and it's the United States, and this is a German scientist, actually, a student of Wieland, who has one of the men who discovered the structure of cholesterol, who's now emigrated to the United States and is asking an even more fundamental question. So I hope you understand what's happening here and can appreciate the questions are getting more and more fundamental as we try to answer a question for a medical reason. What is cholesterol, the substance in the plaque? Oh, it's this 5-ring compound with a little aliphatic tail. I'll show you the structure in a bit, in case you don't know it. And in order to answer that question, you had to figure out how the cholesterol gets made. But to do that, you have to figure out what it looks like as a chemical. And then you suddenly notice that all these other clues are coming from other areas of biology and medicine, completely unrelated, going from plants to people. And you realize that there are certain commonalities of the chemical structures. And those commonalities are actually clues as to how the molecule of cholesterol is put together. I'm going to give you a hint. It's later, we're going to find, many years later, that from discovering the pathway of cholesterol synthesis, how it's put together, that's where the drugs came from, because the drugs prevent the synthesis of cholesterol. So all of this had to happen for those drugs to be developed. But now, in the 1940s and 1950s, we see Fritz Lipmann has actually made a very important discovery. He's discovered how it is that carbon bonds are made in the body. This is maybe the most fundamental discovery, one of the most fundamental discoveries in all of biology and biochemistry. How is energy utilized to activate a carbon molecule so that it can be linked to another carbon molecule? Because I think you all know that the core feature of human biochemistry and any biochemistry is the organic molecules that comprise our bodies that have carbon-to-carbon bonds. And it was Fritz Lipmann who discovered a molecule called coenzyme A that is the key for activating virtually every carbon-to-carbon bond biosynthesis in the body. And he discovered coenzyme A's role in relation, actually, in relation to an unrelated biosynthetic process. But this then introduced the concept that quickly migrated back across the Atlantic Ocean to the laboratory in Munich of Theodor Lunen, and also the laboratory in the United States of Conrad Bloch, who was at Harvard in Cambridge, Massachusetts. Theodor Lunen was at the Maximilians Universität in Munich, and a member of the Max Planck Institute. And what Bloch and Lunen independently discovered, now armed with the idea that coenzyme A, coming from Fritz Lipmann, the idea of coenzyme A activating bonds, carbon-carbon bonds, that was the key clue that enabled them to discover the mechanism of cholesterol synthesis. And so I'm now going to actually show you some chemical diagrams. And what we're looking at here is this 5-carbon molecule, which is that I've been mentioning, this 5-carbon intermediate that is the basic structure that will be made into cholesterol. And it's called HMG-CoA, hydroxymethylglutarate coenzyme A. There's coenzyme A. And what coenzyme A does is basically it makes a high energy bond to a 2-carbon acetate unit that then allows this 2-carbon acetate unit to be added with other 2-carbon acetate units into what amounts to a polymer. And that polymer, in some cases, is limited to 5 carbons, and it's further modified, and is then used as the basic ingredient, adding one after another, to make the core structure of cholesterol and the core structure of rubber, terpenes, the core structure of steroid hormones or of bile acids. So it all comes from this one enzyme reaction. And the enzyme that carries out this reduction of mevalonic acid, the precursor, which comes from acetyl-CoA to acetyl-CoA to HMG-CoA to mevalonate, and then is further metabolized into cholesterol and the related compounds. This turns out to be the key regulatory enzyme. And so if you could inhibit this enzyme, you would then inhibit the pathway of cholesterol synthesis. And actually, that was the discovery made by Michael Brown and Joseph Goldstein in Dallas, Texas, at Southwestern Medical School in the early 1980s, and was awarded the Nobel Prize in the mid-1980s. And they discovered the enzyme, the key enzyme that is the regulatory enzyme that I just mentioned, HMG-CoA reductase. And importantly, they discovered that this is the enzyme that is the regulatory enzyme in human beings that controls the amount of the body cholesterol. They also elucidated a pathway that I won't go through in detail that made it very clear that if you could inhibit this enzyme here with a drug, HMG-CoA reductase, that drug would work to lower cholesterol in the body. And so that then enabled a Japanese scientist whose name was Endo to screen fungi, okay, much as Fleming and others had screened fungi to discover penicillin, now and therefore to develop the first antibiotic. Endo in the early 1980s screened thousands of strains of fungi to identify a few that secreted a molecule that very potently inhibited HMG-CoA reductase, and therefore, according to Brown and Goldstein, using the pathway of Lunen and Bloch, which was based in turn on the discoveries of Lipmann and Lunen, and the earlier discoveries that I've mentioned to you of the structure of cholesterol that enabled all of this, he was able to discover the prototype drug Endo did for what we now call statin drugs. They were further developed in the 1980s by the American pharmaceutical company Merck, and the first was approved in 1987. And so maybe you remember that we saw this dramatic drop in cardiovascular disease, and that a lot of it has occurred from 1980 on. This part of the curve was blood pressure, and this part of the curve are the statin drugs. And the story of the statin drugs, then, I just went over for you. And I hope you can see that more than half a century of basic science resulted in the development of these drugs. Beginning between 1910 and 1920, the structure of the cholesterol molecule, the understanding that the structure must have a very broad biochemical basis that has in common, that takes into common, that includes the biogenesis of rubber. Rubber is a compound which is a direct repetition of these 5-carbon isoprene units. The discovery that these isoprene units are made from acetic acid and made using coenzyme A for activation, and their biosynthesis was uncovered by Bloch and Lunen in the 1950s, taking advantage of the discovery of coenzyme A and its even more fundamental role, an entirely curiosity-driven discovery by Lippmann made in the late 1940s. And this led to Brown and Goldstein discovering through the genetics of certain families of humans who have constitutive high blood cholesterol. They discovered the gene that was important for that, and they discovered that that gene controls HMG-CoA reductase. And then that opened the door for the pharmaceutical discovery, initially in the university in Japan, by Endo of the first prototype drugs, which were then further developed and made manufacturable and synthesized by Merck and Company, and later by every major pharmaceutical company. So the lesson, I think, to take away from this, and I'd like to emphasize to you today that I could be talking about really any field of basic science. The lesson is that basic science can have profound, practical impact. Look at the example of cholesterol and saving lives even today. Where did that impact come from? It came from basic science that was broad, deep, and enduring. You know, if the government had decided, any government in let's say 1920, we know cholesterol is there in plaques, we're going to start a program, and we're going to make this the decade of cholesterol. And we're going to invest $1 billion in solving cholesterol. You know what would have happened? They would have spent $1 billion and they would have had nothing for it. But they could have said something quite different. We're going to spend $1 billion over the next 10 or 20 years or 30 years, and we're going to enable individual scientists, just as you saw here, to follow their curiosity and to go about in their own way to understand deep problems of enduring value, basic problems. How does the body make a carbon-containing compound? How does the body make any compound? And so scientists who were well-funded in programs that were driven by their own initiative, rather than top-down targeted programs, succeeded. Admittedly, it took some time, but they succeeded not just in solving cholesterol, but in many other important advances that were made. And so today, the lesson that I think we all should learn is that funders should invest in great scientists, especially young great scientists with great promise, not necessarily great projects or great goals. The great goals will come from funding great scientists. There are many other examples of that, and a current one, which I won't go through in any detail, is the enormous progress that we're making in cancer. Okay, I think it's no secret that that there are many, many wonderful treatments for cancer today that didn't exist 20 or 30 years ago. And what they're based on is the single most important discovery in cancer biology, in my opinion, which was made by these scientists in the 1980s. And they discovered that cancer is a genetic disease. Sometimes people are born with genes that predispose us to cancer, but more often these cancer genes are acquired by mutation in the body, a so-called somatic mutation of genes that normally function very positively and favorably in the body, but can malfunction when they're mutated to cause uncontrolled growth and metastasis. That fundamental discovery was further enabled by the description of the human genome. In around the year 2000, we had our first complete human genome. It cost billions of dollars to get the sequence of that human genome. Now it's a matter of hundreds to $1,000 to routinely sequence the genome of any one of us. And as a result of this democratization of the genome, beginning around 1920, there are many, many, many genes that have been discovered and connected to cancer. And they've been connected to cancer through fundamental research, not just directed at cancer, but research, including, I have to say, my own research in molecular cell biology, which governs cell division, communication, and physiology. And we still continue to see many new drug targets. And over the last several years, this combined with high throughput methods for drug discovery and the development of monoclonal antibodies has given us really dozens and dozens and dozens of very important new treatments for cancer. And there are drugs and treatments that have been approved that really target every aspect practically of what goes wrong in cancer, whether the targeting is through pills that you take or injectables, monoclonal antibodies that are directed against the cause of disease. Most recently and importantly, there are drugs, a class of drugs called checkpoint inhibitors that are monoclonal antibodies that take advantage of our understanding of basic aspects of the immune system and allow the immune system to be unleashed for controlling, unfortunately, not all, but many cancers. And even as we speak today, we're seeing the development still in clinical trials and often at the university, not yet in pharmaceutical companies, of specific cancer vaccines that are taking advantage of characterizing the genome in an individual cancer and then developing individualized vaccines for the very specific alterations or mutations in one individual cancer. And so we're starting to see bespoke, individualized cancer vaccines that can work today that couldn't work before because we have checkpoint inhibitors that release the immune system and now specific personal vaccines that redirect the immune system according to the nature of the individual tumor. We're not yet seeing the clinical effects of that on a large scale, but it seems very clear that we will. And when we will see the fruits of nearly half a century, starting about the 1960s, of molecular and cell biology harnessed in the most effective way. And looking forward, even beyond that, we have one of the most profound discoveries that was made very recently, between 1910 and 1915, of genome editing, which is allowing us to do genetic surgery. And we've seen the first drug today that has been clinically approved for specific genetic surgery to change a base within a patient in a targeted fashion to correct a very specific, admittedly very rare disease. But we will see more and more of this. And in the decades ahead, in which if any of you become practicing physicians or scientists doing biomedical research, or just living your lives as everybody else does if you choose not to go into science, you will benefit from everything that I've said, from the basic science that has continued to develop And so I went, I've to summarize then, I've sort of taken you through a piece of basic science, which has come, I would say, to full fruition. Discovery of the way the body handles cholesterol and the way the body controls blood pressure. And we've seen, if not the full benefit, close to the full benefit for public health in heart disease. With that success, now after that, cancer becomes an even bigger problem. Why? Because people live longer, and we have seen already, and we are now developing the full clinical impact of 50 years of investment in basic cell and molecular biology, and the further development of tools in the pharmaceutical industry that have allowed new cancer drugs. And that still continues and will continue for the next 10 or 20 years. And I'm suggesting that we are at the very beginning of another era of genetic medicine that has come out as a result of the many methods that are now under development, initiated by Charpentier and Doudna and others, for genetic surgery. And we will probably see the full impact of that as in the fullness of your life on neurodegenerative disease, which is probably the most important frontier. We're making an impact on cancer. We know how to do it. We don't know how to do it in every case, but we know that we can do it. And so that means that over time, cancer will be controlled to the point that, if not in my lifetime, in your lifetime, it will be controlled and can be thought and probably described as curable. But today we have an imposing problem. And I'll leave you with this to think about, because as infectious disease has been controlled, as cardiovascular disease has been increasingly controlled, as cancer, and I would say we're at the midway mark, we can give ourselves a good report card, but we haven't yet controlled it, but we're making progress. All of these diseases will be controlled in your lifetime. And what will be the result of that? People will live even longer. And we're already seeing the result of that. What is the one cell in the body that actually doesn't divide, or only rarely does? And so we have to make do with it for our whole lives. It's called the neuron, okay? And we can't get rid of and turn over all of our brain cells, because if we did, we wouldn't have any memory and we couldn't learn anything. So we're really stuck, okay? So we have to find, what happens then is our neurons start accumulating pathology over time. And we know something about the pathology. We don't know enough yet. There's a lot of basic science that needs to be done. And we're seeing a lot of billion-dollar programs that are directed, just as I warned you we shouldn't do. This is the decade of the brain. We're going to get Alzheimer's disease this decade. We're not going to do that, I'm afraid, unless we really understand a whole lot more about the cell biology of the neuron and the basic biology of the brain and how the brain works. And of course, that's going on, and it's a pressing problem. It's estimated that today, 50%, fully 50% of children born today, babies born today, will develop profound neurodegenerative disorders sometime in their lifetime. And we're starting to see that already. It's very clear. It's becoming a public health problem on an epic scale. So honestly, if you look left and you look right, half of the people or more, probably more, of your friends and colleagues today, half of you are going to eventually develop neurodegenerative disease. And so we've got to do something about this to prevent it or to treat it. And that's going to be your biggest challenge if you go into biomedical research. And it's going to be the biggest challenge of basic research to understand. And probably the answers are not going to come from specific studies, frankly, on neurodegenerative disease. I also suspect they won't come from studies of the brain. They may well come from studies of the liver, when we learn what causes oxidative damage in the liver, for example, okay, or the kidney, or maybe from a plant cell. I really don't know. Nobody knows. We need to invest in curiosity-driven research. And so I have one lesson in science that I think that took me many years to appreciate. I used to think that the scientists I admired were, like incredibly smart, that they had something special going, that they knew some secret that I didn't know. Okay. And then it turned out over time that I became a successful scientist. So for a while there, I thought I must be very smart and I must really know some things that are very special that maybe other people don't know because I made a couple discoveries too. Okay. Now, what I finally discovered, though, is that is the best secret of all. And it's not a secret of any kind. It's the scientific method. Because there was only one really great discovery made in the history of science, and that is the scientific method. It's a way of approaching the world. It's an empirical way of approaching the world. And it was first applied in physics, beginning with Galileo and Descartes and Newton and others, and later applied over and over and over again. And there is no more important discovery or as important discovery that has ever been made. And so if you just stick to the rules of the road and you keep your emotion out of science, which is, of course, difficult to do if you're a practicing scientist, but you can develop the tools to do that, and you simply apply the scientific method, it has endless potential. And it has led to enormous improvements in human longevity, a reduction in suffering, and I believe will lead to the control of neurodegenerative disease in your lifetimes. And I'll leave you with just one thing, a quote. This is a quote from Edvard Buchner. Most people don't know who Edvard Buchner is. I've made a study of him because he's sort of a hero of mine. A German biochemist of the late 19th century, who actually invented the field of biochemistry. He discovered that simple yeast, can ferment sugar when they're dead. You can actually, he discovered, take yeast cells that are alive, grind them up into a pulp, and now as a cell-free extract, they will metabolize sugar as rapidly or more rapidly as a living yeast cell. And in so doing, he discovered enzymes. He discovered the way to discover the method to utilize the scientific method, cell-free biochemistry, to discover how the body builds and metabolizes compounds. And everything that I told you about 20th century biochemistry, the story of cholesterol, but really the entire story of biochemistry, can be traced to his accidental discovery. Accidental discovery, okay? He found that a yeast extract that was made ruinously by accident was actually capable of bubbling up, literally making bubbles that indicated that it was alive, but it wasn't. And so he taught us that you did not have to have a living organism to have the force of life. He taught us that life was chemistry. He taught us, therefore, that life can be understood as a chemical problem and therefore by the scientific method. And he left us with this wonderful statement from the early 1900s that we as scientists should never fall into the trap of thinking that there's a problem that we will never be able to understand, that we shall never know, and we should have confidence that whatever the problem is that we study, whatever the puzzle is, that someday it will be accessible to us as natural scientists, that there is no limit, in other words, to the power of the scientific method. I think that Buchner recognized that and practiced it in his day. I have tried in my lifetime to practice it in mine, and I hope that that you will consider careers in science, and doing so, understand that it's a calling of the highest order that has potentially the greatest impact of anything that we can do. So thank you very much for your attention. I'm sorry I can't be there with you today, but I hope that today's lecture has been of some value to you. in thinking about what comes next for you.